whoops. Okay, when I used to do my postdoc work, we would go out to the field plots, which was about 2 hours away, and we would potato scab research. You can't use farm machinery to harvest the potatoes because it damages the surface of the tubers enough that we have to assess, the disease shows up on the surface, so you can't have any damage to the surface. So one of my first jobs after I got my PhD and I started the postdoc, had to go out into 90-degree weather, super humid, super buggy, hands and knees digging up potatoes in this huge field. Boy, did that give me a humbling experience. So it was luckily a big crew of us, and Dairy Queen was right down the road, so we made the most of it. But you know, this idea that how you harvest or sample, we had to be super careful. We couldn't use machinery. And then we needed to keep things as cool as possible because we had a 2-hour ride back. So we had lots of coolers and ice and putting things on ice. And then bringing them back to the building, we had to put everything into, we had big walk-in cold rooms that we would put them in and then try to get to processing as soon as possible. But before we even did all that, we had to think through how we're gonna do that. And so one thing, couple of things about that is whatever you choose, you document it in detail, because that's really important when you go to publish. Other microbiologists will want to see that level of detail because that helps them assess the data better and look at your conclusions with more background. And that drives me nuts when I go and read published papers and that level of detail sometimes is missing. And it can be very frustrating because you don't know exactly how they went through this procedure. And if you are working in microbiology, it can matter quite a bit what you choose to do for even just your mixing time in a blender. We went through this last time, 2 different soils. They responded differently to what method gave you maximum number of microbes, which is what they were going for in this particular study. They wanted to see that. And then here you're looking at different solutions that you put your soil sample into. And again, you can see it makes a big difference. And so that's why you document whatever you choose, you document that and have that in your paper. Here you're looking at kind of a mock-up trying to get across the point that, you know, when you do here on the top, they're looking at, well, if you decide to so-called fix your samples, anytime you hear the word fixation, that usually means they're going to kill it somehow, dunk it in ethanol, immediately freeze it. And so you have viability sacrificed, of course, and that's a very static type of assay because they're dead. So you're just looking at maybe their genetic profile or maybe just looking at them under the microscope. And then as you go into taking those samples and working with them and keeping the samples alive, then you get these ideas of, you know, incubations and cultivation. Biomarkers are something that sometimes you fix and kill the cells for that, sometimes you don't. And then they're trying to just show that whatever you do, you need to try to minimize the artifacts creeping into your experimental design and process. And so it's just something that you have to train yourself to think about and try to come up with the best situation for an experimental design. This is kind of busy. I don't know, I'm not terribly fond of this figure, but if it helps some of you, it basically, again, is trying to show that when you're at the field site, you have, you know, your sampling going on, and then you have at the field site, you know, you're doing more of the descriptive descriptions, the ecology of it. And then as you bring it back to the lab and start working with it, you're getting into more of the chemistry and the physiology, perhaps the molecular biology. And they're just trying to point out again that the more you manipulate it, the more you can accidentally put in some variables you weren't anticipating. So again, just being very thoughtful about your approach and lots of detail when you write up your methods so that it's all there, well described and characterized. So let's get into a little bit of what you could do once you bring them back to the lab, and especially for your research project in this class, streak and spread play is probably going to be a standard thing that you'll be doing for a lot of you. And so that is something that you've already learned in lab. And there's a lot of information you can get from this, of course. You can look at their colony morphology, their pigmentation. So anytime you look at, like maybe it's a soil sample that you process and do a spread plate, and you get all these colonies on the plate. Every colony that looks different is different. And so you can get a feel for like diversity, like how many different species are in this sample. Just by looking at colony morphology, you can get an estimate of that. And so you're looking at the design of the colony, the color, and is it raised up? Is it sunken down? Is there some kind of pattern on the surface? So you start to pick up differences pretty quickly once you get some experience. And then depending on your question, you may want to go ahead and enrich. So you want to create a situation when you process that soil sample that you're going to maximize your chances of getting your desired microbe. So many of the things that I did as a postdoc, I was looking for Streptomyces bacteria. And so in order to enrich for them, I had to know them. I had to get to know them, get to know their physiology. What do they like? What do they don't like? And then if I'm trying to get them above all others, how do I get rid of the others? And that can be tricky because at least with the Streptomyces bacteria that I worked with, an extra problem was they grow slowly. So a lot of other bacteria that I didn't want grow very quickly, and they could overwhelm the plate and actually grow over my Streptomyces, and that's not what I wanted. So I had to think of ways to process the soil to minimize growth of other microbes. And so if you took a soil sample and you're like, I only want Streptomyces, what might be a simple, maybe a simple way to try to deal with getting rid of the other stuff when you do like a spread plate technique? Can you think of something you could do that might get rid of the other microbes but keep the streps going? What do you do when you need to get rid of some microbes in you? Pardon? Heat yourself up. And that's what we do. We take soil samples and we heat them. And that was one strategy I found that was quite effective. Sounds weird, but it works. You put them in like a drying oven, and you crank up the heat a little bit. And Streptomyces are tolerant, relatively tolerant to heat because they form spores as well. So it was a good strategy for us. It worked. And so that was one strategy we did. And then if you're like sick with strep throat, what does the doctor give you? Yeah, antibiotics. So we would mix into the recipe of the agar plate antibiotics. And we knew which antibiotics most of the streps were not sensitive to. But many other microbes in the soil were. So we were able to concoct a recipe that had like 4 different antibiotics that weren't harmful to the Streptomyces, but killed off a lot of the others. So we hit it with kind of a multiple approach. Heat the soil. First, we just dried out the soil. We left it on the counter with cheesecloth over it and let it dry out, because just drying out, if you limit water, I mean, every living organism needs water. So if you start drying out something, you're killing off certain things. And again, the streps form spores, so we weren't terribly worried about that. So dry out the soil, heat it a little bit, put antibiotics in the spread plate. So you can see we were trying many different things to try to enrich for the Streptomyces. And then we also got to know them physiologically, like what kind of carbon sources do they like? What allows them to grow as quickly as possible and hopefully outcompete other microbes on the plate. And so we designed different media. I had to play with that a lot, trying to find different recipes that would work. And that took a fair bit of time. And so differential media, what I was just talking about was more selective media. So where I had to figure out their physiology and optimize their growth on the plate. Essentially, differential is the idea that you create a recipe for that agar plate that when the Streptomyces show up, you can easily identify them, hopefully just visually. And so differential means you can tell the difference between Streptomyces in my case and all the others. And with us, Streptomyces bacteria are pretty unique anyways, so we didn't have to worry too much about a differential media because they form colonies that are pretty distinct from the other bacteria. So it was pretty easy to figure that out. But one thing we did do when we were hunting for the potato scab pathogen, that one can look like an ordinary Streptomyces. It's actually a Streptomyces bacteria that causes potato scab disease, but there's a bunch of other Streptomyces that can kill it. And so we had to tell the difference between the two. And we're like, okay, how do we tell the difference? And if you pop tryptophan into the media, for some reason, that pathogen likes to take tryptophan, and as they grow, they spit out a pigment that's nice and like almost black. And so it shows up around their colony. And so again, very nice visual. That was very handy for us because we didn't have to do any expensive procedures, and we were able to look at that plate and go, oh, that looks like a pathogen. And that one looks like a non-pathogen just based on the pigment that was being produced. So there's tricks like that that you get to learn as you learn more about your microbe and what you're going for if you're trying to target and get a certain microbe from your habitat. And then you get all these colonies on the plate, which can give you a lot of valuable information. You can do diversity, like I said, everything that looks different, all the colonies that are different. So you can tell diversity of organisms. And then you can also get estimates of population sizes. So you can count the colonies. That's when you did your spread plate exercise and you got all those colonies. You know, we could have gone through and counted them all up and then figured out what was in the initial sample we gave you. And so you can get estimates of population. So you can do population estimates, you can do diversity estimates, just from doing the spread plate technique in particular. But if you start working with these bacteria or fungi and get them onto agar plates and they're all pure, that's great. It can be very helpful for certain things, but you have to, of course, realize that if they're in pure cultures, you're not really thinking about community dynamics going on. And there's other things that you can do for that. So phenotype, very often it's the spread plate technique. You dilute your sample typically and start looking at colonies. There's some other techniques that you can do. You might want to take a soil sample, let's say, and just say, you can, through lipid profiles, you can actually learn a lot about who's there and even a little bit about their physiology. Like, are they stressed? Are they well fed? Just by looking at their lipid profiles. And so what this zooms in on is the cell membrane. So we zoom in on the cell membrane lipids, and you can extract them and then analyze them using gas chromatography. And so FAME, I'll go through that. And then PLFA, I think I also mentioned that, phospholipid fatty acids. So you're looking at the idea that you can extract these cell membranes, and remember now, phospholipids, that's the cell membrane. And so they have fatty acids on them as part of the phospholipid. And so fatty acids vary. So microbes make different types of fatty acids depending on their environment and what's going on inside them. And so you can use that information, compare your samples to big databases out there to get a feel for what's going on just by looking at lipid profiles. You can even identify down to species level in some cases if the database is big enough. And so you can actually just take a soil sample and run these types of procedures and get a pretty good idea of who's there, at least what groups of microbes are there in that community. So I didn't do these as a postdoc myself, but a fellow postdoc in our lab was doing this quite a bit. And it was, so I kind of shadowed what he was doing. And just remember, phospholipid bilayers, this is like the cell membrane idea. We're not focusing on the proteins that are embedded in there, but just recall that you have these fatty acids that are dangling, so to speak, off the phosphate group. And you have, here's a more detailed description of the phospholipid. So again, you can see the fatty acids are carbon and hydrogen. There's that little oxygen there. And that's the hydrophobic portion of phospholipid, and then your glycerol and phosphate are the hydrophilic. So hydrophobic region, hydrophilic region. And then, of course, we're ignoring the idea that there's all those proteins in there. That's not what we're going for, but of course they're there. So then, depending on the microbe and their habitat, they can vary what type of fatty acids are in their cell membrane. And that's key because we use that information to get a better feel for who's there, what kind of state they're in physiologically. And so here you're looking at omega-3, omega-6, omega-9 type fatty acids, which is just referring to where the double bonds showing up in the fatty acid tails. And I'm sure you must have heard a little bit about that just out in the real world, thinking about diet. You know, they say certain fatty acids are good for us, others not so good. Do you remember which ones are good for, well, better for us, I should say? It's on here. No, actually, we need omega-6s in our diet, but the American diet, the standard American diet, which abbreviates down to SAD, standard American diet. It's sad. So true. Omega-6 is too much. We get too much of that. And so we're shifting out of balance. We get way too much omega-6 with the standard American diet compared to omega-3s. And so most medical doctors would agree that Americans in general need more omega-3s in their diet, which is not always easy to get, especially with your food choices. So I actually supplement with omega-3s. I take algae created omega-3s myself because don't have the best diet currently. So I hedge my bets on that. But you can just see that there's all kinds of options. And this is just a little snippet of the different kinds of omegas, that 3s and 6s and 9s and all the different fatty acids. But basically what you're doing is you get your soil sample. We typically try to, very often we are hunting for a particular type of bacteria and then trying to figure out what's going on with it. So we work with a pure culture and pop it into a broth. The one thing with this technique is you have to follow the standard procedure to the letter. And they have like a documented, this is what you do when you want to extract fatty acids, because it needs to be consistent to the database on how they extracted everything to get the database of information. So you have to be very careful of following it to the letter. But basically you grow up your bacteria, you extract the fatty acids, do a little bit of chemistry wizardry, and then you run it on the gas chromatography. And it spits out the output, and that can help you compare to the database and get you some information, including possibly identifying your organism. So we were doing this when we were looking at our fields of potato, the soil, the potato that we were putting potatoes into. We wanted to get an estimate of how much is the pathogen there. And then we were trying to look at how much of the pathogen is there that was attacking the potatoes. So from that information, you can get a lot of different pieces of information. So here, if you extract, and they're denoting that it's intact polar lipids, here's the phospholipid fatty acids, they wrote it out there. They know that those are telling you have viable cells. And so again, when you take a soil sample, sometimes you want to know how many are living and how many are dead in this soil sample. That might be important information. Metabolic status is another thing. You can actually get a little bit of look into, are they stressed or are they well fed? Here on the neutral lipids, if you extract those, sometimes that's telling you if you have sterol type lipids. Bacteria don't produce sterol type lipids, but fungi do. And so that might give you some information on your fungal population. If there's diglycerides present, that's telling you you have non-viable cells, they died off. And then just by looking at some of the quinones that you isolate, you might be able to tell a little bit more about their physiology. So again, this is just trying to get you to think about the idea that you can really get some good information from this. This one going off to the left, the total lipid alpha ether lipids may just sound like mumbo jumbo, but it's really archaea, the domain archaea, that group of bacteria or prokaryotes, I should say, they produce ether linkages when they are creating their cell membrane, and bacteria don't do that. So that's a huge difference. And so you can key in on that, and you can actually just say how many archaea are there. And what you do is you hunt for the ether, the alpha ether lipids, and that will tell you. And then here off to the right, you're looking at this lipopolysaccharide, lipid A. That's unique to gram-negative bacteria. So lipopolysaccharide is often abbreviated LPS, and gram-negative bacteria have that, gram-positive bacteria do not. So again, it's a nice way to get some information on that distinction, if that's important to what you're doing for your research. So it's kind of weird, but lipids can be very helpful. And it's not terribly challenging to do that. And the database you can compare it to has just gotten bigger and bigger and bigger over the years. So it's quite robust. And then, of course, molecular. I mean, I think most of you would certainly recognize that idea that you could extract nucleic acids and just do the DNA sequencing. And then you know a whole bunch about the microbe, which is true. And you can actually just take like a soil sample and then not do anything with it yourself. Just take the soil sample and send it off and say, sequence everything in here, all the DNA, and tell me who's in this soil sample. And the gene sequencing has gotten that good where they can extract DNA from the soil. Now there's millions of microbes in that soil, and yet they have the fancy computers and algorithms to actually spit back data telling you exactly who is in that soil sample, down to the species if you want it. And so it's pretty powerful now. It's great. It's a really great tool now for microbial ecologists. Not too long ago, you couldn't do that. They didn't know how to extract DNA cleanly from soil. Soil is very dirty, messy, complicated, complex. And so getting DNA from that and not have it just like totally fall apart in the process, they didn't know how to do it until fairly recently. So processing it, now they have this down, but it took a long time for the scientists to come up with how do we take a sample and process it well enough, but not too much, so that we can actually extract a good DNA. And so it's a lot of use beaters or blenders. Sometimes you use chemicals to lyse. You have to lyse open the cells. Some cells are harder to break open than others. You might have to do phenylchloroform extractions. You might have to do some ultracentrifugation and then some chromatography techniques. But sometimes you can do some of this yourself and then send it off and have them finish the sequencing. Sometimes you just send the soil off and then, or whatever, and say, just do it all, and they can. So one of my grad students who's now working in the indoor egg program, Carrie Farkas Lasich, she was trying to extract DNA from endophytes that lived inside orchids out near Munising. And this was a little bit ago, and finding a good method to extract DNA from fungi, very challenging. They're harder than bacteria even to try to do this successfully. So she went through a lot of troubleshooting to try to figure out how to extract DNA from fungi. And then she finally stumbled on a published paper that had a method. So, you know, the companies would send her these kits and say, use our kit and it'll work, and it wouldn't work. And so she's like, ah. So she started going back to some of the more older processes, kind of the old school methods. And then she finally found a paper that was old school method, but she got it to work. So it was quite a process for her, and it took quite a bit of time, but she got it to happen. So here's a flow chart, and this is some options of what you might be able to do with your sample in terms of the molecular aspect of it. So if you've taken Intro to Cell and Molecular, some of this might look somewhat familiar. we won't be doing this in this class because this is not a molecular biology class, but basically, so many of these methods are pretty powerful in terms of being able to get clean DNA, sequence it. Omics, have you heard that phrase? The very last word here, omics. That's like proteomics. What's the other one? Metabolomics. The proteomics is looking at all the proteins that are made by the microbe. Metabolomics is all the metabolites being made, which is actually becoming a thing. You might start seeing, I've already started seeing it in supplements you can buy, that you look on the ingredient list and they'll say, how do they often phrase it as fermentation products of whatever bacteria. And so that's basically you're ingesting metabolites. And so you can actually assess that. You can say, what kind of metabolites are these microbes making? And it's usually a lot. So the omics idea kind of get at, in totality, you're looking at, you know, all the metabolites or all the protein being made. So very often, hunting and working with the messenger RNA and isolating that is desirable, not just the DNA. So messenger RNA, you go back to the whole process of how you work through transcription and translation. You have your DNA, and then you do the transcription event, which is getting your messenger RNA. So basically that step is saying what genes are getting turned on and allowing for messenger RNA to be built off of it. So you're looking at, if you get your messenger RNA isolated, you're answering the question, what genes are turned on? What are the microbes doing in that habitat? And that can be super important. So very often you'll see that method being done in papers where they're focusing on what are the microbes doing in a particular habitat. They'll isolate the messenger RNA, and then you can, if you want, you can use reverse transcriptase, RT, reverse transcriptase, and make a copy of DNA off of it. And then you can use that DNA and assess it further. So it can be very valuable. It's tricky. I used to isolate messenger RNA out of mycorrhizal fungi, and it's tricky. It's gotten better, but back then it was super expensive. So, and it was one of my committee members' grant money that he bought the kit for me, and yeah, I was sweating when I was running that experiment, because if I didn't do it right, that was a lot of money down the drain. So luckily it worked, but that was a little bit nerve-wracking because it was really, really expensive. So this idea of you can isolate the DNA, you might want to isolate the messenger RNA and look at that directly, or turn it into DNA and do something further with it that way. You have a lot of options. And, you know, when you look at asking the question, what are they doing physiologically? You don't always just have to go for the messenger RNA. That's like the gold standard. That's what I would pick, but sometimes you can do other things too, to say, hey, what are they doing? You can look at the ribosome content of the microbes, and that can give you some information too. And then some of these others too are looking at the intracellular energy reserves. That's just, you're looking at how well fed are they, essentially. So poly beta hydroxyalkanoates, what a mouthful. Those are, a lot of microbes make those as storage molecules. So if they're really well fed and having like feast conditions, they'll have a lot of that. They'll have a lot of ATP. So you can assess those in order to determine how well fed, or, you know, are they in good times or are they experiencing tough times? So there's other strategies that you can use to ask the question, what are they doing physiologically? By the way, it was some magazine. Oh, it was a travel magazine, and they were talking about hotel rooms and how hotel rooms test for microbes being present. And I really hadn't given that much thought, but the way they do that these days is they have little kits very easily testing for the presence of ATP. And I'm like, huh, what? But it makes sense because microbes work off of ATP. And so if you swab a surface and pop it into the kit and it pops a certain high number of ATP molecules, then the assumption is there's a lot of microbes there. Apparently that's the standard these days. A lot of like hotels and other places, if they're trying to keep tabs on how cleanly their surfaces are, they do it in like, I don't know if they do it regularly in the food industry, like in the kitchen area, I don't know. But I thought that's a pretty slick method, pretty easy. Anyways, you can also look at physiology through gas production. So when you take some gas samples, these are hollow cylinders that you put into the ground to a certain depth, and then you cap them with these rubber caps, and then they have these little ports in here that you can stick a needle down, and then you can like use a syringe and extract out some gas from that headspace above the soil, and then take that sample back and analyze it chemically. And so Dr. Van Grinsven does this in the soils class, I know, and he's done that with some of my grad students. He's helped out trying to figure out what's going on. And very often we're targeting carbon dioxide. We're looking at that. But using that idea as kind of a proxy, if there's a lot of carbon dioxide, that usually means there's a lot of respiration going on by all kinds of organisms. We're not just targeting microbes. There's all kinds of creepy crawly things in the soil. So you can't just say exactly where those gases came from, but it is indicative of how active that area of the soil might be, for example. Physiologically, how busy are they? Are they alive, or are they in a resting state? Are they in spore state? Stable isotope analysis. I'm not going to really get into that, but that is another powerful technique where you can use those ratios to look at a little bit more about what's going on in the soil physiologically. Microelectrode measurements, I've never done that. But they get at looking at some of the physiology through that. So this is kind of lots of kind of very different ideas being listed out here. But again, just trying to show that depending on your question, especially nowadays, the technology has gotten so much better that we can now we can answer questions that we couldn't even like a decade ago, in terms of the technology just wasn't there. And it was a nice question to ask, but we didn't have a means to actually get at the correct data to try to start assessing whether or not that question can be answered in any significant way. So lots of ideas out there. I'm going to shift into the second slide set. Couldn't put it all on one slide set. Educat got grumpy with me, so I had to split this out. Okay, let's see if I can. So this term you may have heard, metagenomics. Another way to think about it is ecological genomics. I think that's a better way to think about it, but they wanted one term, so they went with metagenomics. So what you're doing, what that term means is, let's say you take a soil sample and then you just send that off and say, tell me all the DNA that's in here, get it sequenced out and get it all put together into a database of who's there. You can actually get not only who's there, but population differences. You can find out if a certain microbe is dominating that habitat and another set of microbes is very low in population. So you can get relative amounts. You can get all the way down to the species level if you want, but that costs more money. But basically you're examining all the genes in a habitat in that sample that you're taking. And so, like I said, you can just take your soil sample and send it off. You don't even have to do some of the prep ahead of time if you don't want to. Price goes up if you do that, but you can do it. And so basically, I'm not gonna expect you to know the step by step. This is not a molecular biology class. The big picture idea here is that you can work with a raw soil or water sample. You don't have to grow up the microbes in the lab. You don't have to do that. In fact, you don't really want to do that because a lot of microbes in that water or soil aren't going to grow up on the agar recipe you picked. And so just sending this off and saying, tell me everything that's there, how much of everything is there. And they can do all of that and more. So they can look at also expression of phenotypes. So that's getting at, maybe they're going to start extracting the messenger RNA and working with that. So there's different ways to do it. You can actually use E. coli as kind of a workhorse in this process. You may have done a little bit of that in Intro to Cell and Molecular. E. coli is basically just being used to make many copies of the DNA you're after. So if you're after a specific thing in that sample, you could use E. coli if you know how to work the process. And basically E. coli is just making many copies for you. They're just a workhorse in the pipeline. So you can capture lots of DNA. So BACs are bacterial artificial chromosomes. So there's all kinds of fancy techniques, which you don't have to memorize, but basically you can devise methods that they have now where you can do, there's a lot of sophistication now to what you can do with that sample in terms of how to process it, depending on what your question is. And so large and small pieces of DNA, no problem. And it used to be a real problem. If you had large intact pieces of DNA, that was difficult to work with. Small ones are pretty easy, but now that they have bacterial artificial chromosomes as part of the process, it's not so bad. But basically here, I'm just trying to show you what would they do it for, like how, under what conditions. And so you can see someone, Venter et al. Oh, Craig Venter. He did the Sargasso Sea. So Craig Venter was one of the business people who owned a huge business that helped the government with the Human Genome Project. So he was the private business. It was a private government collaboration. And he's very rich. And so he was able to help sequence the human genome. And so that was that's like his claim to fame. After he finished doing that, he decided he wants to find out more about our oceans. And so he bought a huge research vessel and started going into different areas, different oceans and sampling. And in this case, it was the Sargasso Sea and Panama Canal to the Eastern Pacific Ocean. And apparently he did a lot of shotgun cloning techniques. And again, this is just trying to get you to realize that different techniques can give you some good information. There's different strategies out there, but I'm not going to get into the details. So Craig Venter is actually very interested now in microalgae and algae in particular, just in general, but microalgae. And he is of the opinion that algae are going to become a dominant part of human life in the sense of we'll be eating it. It will be used for biofuels. He's very excited about algae, which I think is great because algae are worth getting excited about. So for someone of his prominence to throw a lot of resources and research toward it, I think is good. It's a good thing. So here are some of by the way, I have a slide set that gets at some of these definitions if you're interested, but I'm not going to test you on them. I'm not going to say, give me the definition of a cosmid. I'm not going to do that. But if you're curious, I do have a slide set with some definitions here. But basically, it's just having you kind of look through this in terms of what they might be doing with this technique. Like what kind of information are they going for? That's more what I want you to look at when you look at these tables, because it can be very varied in terms of what they're going for. So these techniques are powerful in that way. It allows you to answer a lot of diverse questions. Here is just a visual kind of getting at this idea of large versus small pieces of DNA, and here they're honing it into artificial chromosome process, or just doing shotgun cloning, which is a little bit more straightforward. And so again, it's just technology is great in this day and age. It's very exciting. There's a lot of options out there to use and get some really good data from that. And so you start with your question, and then you start thinking about, okay, how could I assess that? And then, of course, the big thing that it all hinges on is how much money do I have? So if you don't have the money, this may not be an option. It's come down in price, but it still costs some money. Okay, so sometimes you want to quantify, you want to count how many are there. And the tried and true method is to get out your microscope and start doing some estimates looking under the microscope. So direct counts, that would be a direct count strategy. And that is probably it does give you the highest numbers of microbes compared to doing a spread plate and counting colonies, because you've biased your sample, because not every microbe loves that recipe you developed for that agar plate. So only a fraction of the microbes are going to grow from the sample. Whereas if you pop them onto a slide and look at them, you can get, you know, all kinds of other microbes that wouldn't grow on your recipe. You can't always tell if they're living versus dead, though. So if that's an important question, you have to do some special staining to get at that, which you can do. There's stains out there that aren't too terribly challenging. And so the other idea is not just living versus dead, but who is dormant or who is active? That's another question. And so that's another thing that you have to, and there's stains for that. You can look under the microscope, stain your samples, and you can tell which ones are active and which ones are dead or dormant. It can be tricky. I don't like doing this with soil samples. Soil samples are kind of a nightmare. Diluting those out and looking for things, it's not fun. I would not choose it. If you're going to do this, you actually use a special microscope slide. It's not the type that you use that, for your gram stains, for example. It's different. And I'll show you a picture of it. Another strategy you could do, which I would pick hands down, is either the Coulter counter, or my top pick would be flow cytometer. That's like the gold standard. And flow cytometer, there's one in the hospital, and one of my previous undergrads, she didn't get to use it. They wouldn't let her touch it, but it's very expensive. But she got to watch the technician put her samples through, and it's amazing. It'll count tens of thousands of cells in just seconds. So it's so nice, but very expensive. So this is the fancy microscope slide. This is called a hemocytometer. What's the other word for it? Hemocytometer. I have it. Well, Petrov Hauser chamber, named after the people who devised it. But basically, it's a fancy slide with some wells etched into it, and you pipette your sample into the well. And then there's this microscope cover slip. There's a microscope cover slip here with a grid on it. So your sample wicks under that. And then as you look under the microscope, you'll see something like that in the middle. And it'll tell you how many of these squares, how many of those you need to count the microbes in. And then it'll give you a formula to calculate your population size. And so, however, it's like, oh yeah, you can probably count about 200 cells per minute accurately for the technician. That's not a lot. And, you know, if you have a technician doing this all day, the chance for error understandably goes up. Your eyes get tired, you get tired. So this is something I do only for very small experiments that I'm doing, where I don't have to do this all day. I'm just kind of assessing very quickly. So I don't really depend on this very heavily. The Coulter counter is a machine that you put your sample in. And again, I'm not going to ask you to know the theory behind it, but it it'll count the flow here, and you can get some data that way, but it's not really good at picking up small cells, like really small microbes, so that's a drawback. Flow cytometry is the best. I would pick that. And so this is using laser beams, 70,000 cells per second. Yeehaw, that is awesome. So this is the gold standard, and I would definitely pick this if I had a big experiment where this is a big part of my method collecting. Really good. It even can sort your cells. It can detect viability, biochemical properties, and somewhere on here I thought I had that it can sort. It can sort. It can actually sort your cells by type, depending on how you process it through here. So really, really good strategy if you have to get a lot of this kind of data. That didn't show up too well on the back, dark background, but here's another picture. Here, oh yeah, they're sorting the samples here. So you can even do that if you want. So go for that. Unfortunately, super expensive piece of equipment. So you need to have somebody that has this, and you beg for use of it. So, all right, we'll stop there and kind of wrap up next week.